So whenever you're ready.
All right. Hey guys, welcome to the December SNHS meeting. Before we get started here, I just want to remind you guys to all log into our website at www.frhssnhs.org. Just because there's a lot of important announcements there, and if you want to do anything SNHS related, it will be done through that website. So just to give a quick overview of what we'll be covering in this presentation today. So some announcements are that we're going to be looking for guest speakers to speak during an SNHS meeting. Vote for charities. We're going to be donating to a charity, and we want your guys' input on what charity to donate to. So there's a survey on that in the remind. We have a new good standing system, T-shirts, and then also hours requirements and reminders on that. And we also have a ton of new volunteering opportunities for you guys to get hours, such as the science fair, applying for internships, summer programs, scholarships, twist submissions, trivia night, science memes. And then we'll also be doing a This Week in Science. And finally, tutors, keep watching for some additional info and updates coming up soon.
Okay, so we're just going to be looking for guest speakers. If you know anyone or have anybody that would be interested in speaking to SNHS during one of our monthly meetings, please go to the hours opportunity section of our website, which I linked below. And there will be a Google form that you can fill out where you basically just detail the information of the person that you think could be a good fit for speaking. And this can be a parent, employer, mentor, anybody, as long as it's science related and kind of goes along with our mission. And please, if you have any questions, feel free to reach out to any one of us.
Okay, guys, we are donating to a local, hopefully, charity with some of the leftover money we have this year from your fees. So check out the survey in the remind and get your voice heard. Okay, the new good standing system. I spent way too much time on this, so guys, please make use of it. Basically, each of you has your own file, and it's private to you, and only the board can see it. So DM me if you see mistakes, and just follow the hyperlink on the good standing page with your student ID number.
Okay, so everyone, you need to order your t-shirts. I'm super excited for this. We're making it due by the end of the semester, so get that done, and it's going to be super fun.
Okay, just a reminder about good standing and such. We lowered the requirement to 5 hours this year, so that should make things easier on you guys. There are no event requirements, so you don't have to worry if you completed everything in one event or something. And to those of you who ordered t-shirts, you will be required to pay that fee to remain in good standing. And to those of you who have not paid your membership fees yet, please pay those. Thanks.
Okay, so we have what I hoped to be a December event, although depending on who signs up for it, it might be pushed back to January. And it will be a science fair. So we're looking for teams of 3 or 4 of you, and we want you to come up with an experiment or something that interests you. You can kind of create a video about or a presentation. And you can see some ideas here, chemistry or our chemistry, physics, ecology, stuff like that. You do not have to go with one of these suggestions if you have your own ideas. There will be some prizes, and it is worth 3 hours. So if you guys are struggling with getting hours, this is a really good way to almost be scot-free on that. The sign up is here, and I believe it should be on the website as well.
Okay, so a super exciting event that we have coming up super soon is this year's virtual trivia night. So Ryan Zhang put together this amazing format, and we're going to have tons of questions. I think about 150 questions and lots of different sections. There will be prizes for the winners. That's super exciting. You have to sign up before December 11th, though, so get on that. Also, there is a maximum amount of teams that can sign up, so it might even fill up before then. So basically, sign up ASAP. You can sign up at trivia.itsryan.org. It will be taking place on December 17th, approximately from 6:30 to 9:30. There will be tons of great prizes that we've been figuring out. And you can get hours for tons of different honor societies, not only SNHS, you can also get hours for Mu Alpha Theta, NEHS, NHS, Tri-Am, and Key Club. You can do it in a team with 3 to 4 people, friends and family and Fossil alumni, all welcome. And you can get together with your team virtually.
All right, cool. So there's another hour opportunity for 3 hours, another if you're accepted, to apply to summer programs or internships. So we have a big list under our science website under hours opportunities, such as there's some from New York, Telluride. There's an Academy for Math and Science, Clark Scholars opportunity. They range from costing a few $1,000s to being completely free. So you'll just pass a screenshot or of your confirmation email to your hours log in order to have some proof. Plus, if there's others that you discover that we don't have listed, feel free to apply there and do that.
So we miss you guys and we want to hear from you. So if you have been thinking about something cool in science, or you read a cool article and you want to tell SNHS about it, feel free to fill out the form under hours opportunities on our website, and we will give you an hour of volunteering, and we'll put it in our next meeting. Another thing, we want your memes. So if you have 4 science memes that you'd love to share, submit it to the hour log form under proof, and we will give you an hour of volunteering. Our favorites are going to go on the website.
Okay, now we are going to be presenting our This Week in Science. And for those of you who have made it this far, the password to our monthly meeting is holidays, plural, all lowercase. So HOLIDAYS.
All right, hunger and loneliness, a surprisingly similar mechanism. So this study was done at MIT, and a researcher and several of her peers fasted for 10 hours and then were presented with several images of food like pizza and chocolate cake. And they found that dopamine producing neurons in a specific area of the brain lit up. The same people the next day underwent 10 hours of isolation from people, social media, you name it. And that evening, neurons in the same spot activated in response to people chatting or playing team sports. So a couple of additional findings they had were that generally lonely people had less of a reaction, and the reactions happen even when people aren't hungry or lonely, but they spike when you are. So big takeaways: eat when you're hungry, it'll make you happy. And even if you're physically distancing from your friends and family, take the time to connect with them. And I know that FaceTime calls suck, but it'll probably make your brain a little happier, so worth it.
Okay, so my This Week in Science is on a research study that studied how COVID-19 harmed the brain and its functions. So we all know that the physical symptoms of COVID-19 are pretty well documented and researched, and those are easily detectable for the most part. But researchers in the UK have found that COVID-19 can also really significantly affect neurological functions. And they found that strokes, confusion, and psychosis were increasingly common among people that were hospitalized with COVID-19. Specifically, it found that 77 out of 125 COVID patients experienced a blockage of blood flow to the brain. And blood clots are already something that we've seen as a symptom in COVID-19, but just not in the brain, which of course can lead to strokes and other problems there. And then also a third of the 125 patients also experienced other problems, such as a shift in mental state, including confusion, personality change, or depression. But that most likely appeared from quarantine or social distancing or social isolation and things like that. Um, so it's pretty clear that the virus does have effects on the brain, but more research is going to need to be done to determine how exactly COVID-19 affects neurological functions.
I'm sure you guys have seen some videos of this giant guy collapsing, which not only was it devastating to see, but it was a huge the giant radio telescope. It was huge for astronomy as a whole. So that little thing that's supported by the wires, little it looks like, it's 900 metric tons. So it is super old, but it does have some critical hard to replace features. It was the largest from when it was built in 1963 until 2016 when China completed their project. And it had incredible sensitivity, almost unparalleled. So it was able to sense, this pulsars, right, spinning things in space. One of the best out of all from pretty much anywhere around the world. And as transmitting radio waves is a unique thing to telescopes, as not most telescopes can only receive radio waves, while Arecibo was able to use it to bounce waves off planets, but especially for its original use of utilizing it to study like the composition and different parts of the ionosphere. But even though it was really sad to see it collapse and the loss of these features, a lot of the crucial discoveries that are from when they go back, reanalyze the data that's been found, as sometimes it wasn't clearly seen at one point, but with new discoveries, you can kind of interpret the data a little more. So the data that's been collected by this telescope is able to help create new discoveries, hopefully.
Okay, so a group of researchers at Harvard Medical School figured out how to restore the vision of mice whose optic nerves had been damaged. And this is huge. So a little bit of a background on how DNA regulates aging. So if you don't know, the process of aging is actually a biological mechanism we've evolved with, because individuals aging and then dying off is actually beneficial to the species. The telomere is those little tips of each chromosome in that picture. Telomeres don't code for anything, but they shorten over time, and when they get too short, the cell can no longer replicate, and that causes aging. The other mechanism of aging is methylation patterns. And you'll see in this picture up at the top, there are methyl groups, which are the CH3 molecules here. They're attached to the DNA in certain parts, and they tell transcription proteins where to which genes to code for. So certain enzymes have the ability to influence these factors. And the researchers injected some of these reprogramming enzymes into the neurons that were damaged in a very specific viral gene vector. So they do this by basically splicing the human gene into a viral genome and then having the virus infect wherever they want to put the gene, because the way viruses work is by making the cell code for their genes instead of its own. And so in that way, you can use viruses as a vector for gene therapies. So they were doing that with these reprogramming factors. And there are 4 of these reprogramming factors that the mice use to change methylation patterns. And if you use all 4 of them at once, it causes cancer. So they only used 3 of them, which is perfectly healthy. And the way they did this experiment was by intentionally damaging the mice's optic nerves first so that it simulated aging, and then doing this viral gene vector, gene therapy vector, to kind of regrow the nerves. And what they found was that even if the damaged ganglion cells were dying or really damaged, the injection prompted new axons to grow and reach back into the brain. And so in visual acuity tests, which is like kind of like what you do at the optometrist's office, where you look at numbers or letters and see if they're blurry or not, they do the same thing for mice. Obviously, it's not the same test, but they can tell the visual acuity. So when they damaged the optic nerve, the visual acuity of the mice went down by like 25%. And after they injected the virus with the reprogramming factors in it, the visual acuity of the mice went back up to where it was when they were super young. And what this experiment means is basically that we've been looking for ways to reprogram our genome for years and decades, because we've known for a long time that DNA intentionally ages. And maybe we shouldn't mess with that. Maybe we should. That's a philosophical issue. But they have this experiment basically is suggesting that we might find a way to do that. This experiment in its own isn't a way to do that for humans, but it is kind of the lead into it.
Hi, This Week in Science is about a new study that found out that alligators can actually regrow their tails. So everyone knows that small reptiles like lizards and like geckos and those kind of critters can regrow their tails, as well as some limbs, I'm pretty sure. But now a study from Arizona State University found out that young alligators can do the same thing. And they can regrow up to 3/4 of a foot of a tail, which is about 18% of their entire body length. So that's a significant amount of tail. And their regrown tails are actually structurally complex. And they contain a skeleton made out of cartilage, and they're surrounded by various kinds of connective tissue and veins and other circulatory material and stuff like that. They found out that the tails are regenerated in a process that is part regeneration and also part healing, so they kind of work together. And this brought up the question, can dinosaurs regrow their tails too? Because alligators and dinosaurs share a common relative. So is it possible that there could be some fossils out there of some dinosaurs that have regrown tails? I mean, I guess we'll find out soon enough. And when was this trait lost? For example, we know that birds can't regrow limbs and tails and all that. So when was this trait lost? So that's the question. Also, further research regarding this topic of limb regeneration and tail regeneration could lead to developments in new medical therapies to treat injuries and diseases like arthritis. And I got this source from ScienceDaily.com. They partnered with Arizona State University.
Okay, so my This Week in Science is about a newly approved drug that can actually prolong the lifespan of individuals affected by progeria. So just a little background on the disease. Children with Hutchinson-Gilford progeria syndrome, or progeria for short, often die of heart failure, heart attack, or stroke before the age of 15. So a newly improved drug called Zokinvy is the first and only approved treatment for progeria and certain related symptoms. So kind of just a background on how progeria affects these affected children. So children with progeria have higher amounts of a defective protein called progerin, and this protein gets stuck in cell membranes and can't be recycled for fresh proteins, ultimately causing the cells to prematurely age and causes the blood vessels and connective tissue to become stiff, which, as you can see in the picture, kind of presents as this like elderly look for these young children. And this newly approved drug actually causes a mutation in their genetic code, which interferes with the production of this progerin protein. So it kind of just not only prolongs their lifespan, but also kind of decreases this like elderly look that they have with this disease. And with this drug, it's not a cure because with it being ingested orally, it doesn't fully affect the genetic code to the extent where it'll totally stop progerin production, but it has just decreased it to a level where they're able to manage it and just extend the children's lifespan.
Okay, and that's it.
That's the end of our meeting. Thank you guys for watching the video, and make sure to check out those links. This slideshow should be up on our website, and if you have any questions, just email us. Yeah, hope you guys have a good rest of your December, and have a good winter break. And now I need to stop recording the meeting.